Thank you very much, Toon, and thank you to the organizing committee for the opportunity to be here. I guess we'll talk about the tiny fungi now to a certain extent. We spent, as Toon mentioned, the last 2 days discussing various aspects of Ascomycete yeast taxonomy, and came away, I think, mostly satisfied with the results. Very little bloodshed and pretty much cooperation, so that's always nice. So what I'm going to do is give you kind of an overview of the yeasts. It's sort of like one of these tours of Europe where you're in Belgium in the morning and Netherlands in the afternoon kind of thing, but we'll keep it moving. And the same goals for yeast, I think, as any other group of fungi. One of our goals for using molecular methods especially is to identify species, and we've seen an explosion of new species in a relatively short time. We're trying to circumscribe genera and families and try to understand the evolution of yeast. But another aspect of this comes out as we begin to group species based on phylogeny. I think this allows us the opportunity to better predict the expression of certain phenotypes that might be biotechnically important, such as particular groups of chemicals, for example, or maybe understand genetic relationships in terms of genetic mechanisms a little better. So I think this is something that's an opportunity not only for us, but maybe a selling point, especially to the biotechnologists who are looking for something, and if we can get a hint, we might go into a particular phylogenetic group and be able to help them a bit find something new. For the yeast, we've had relatively good luck on recognizing species from the D1, D2 domains of large subunit ribosomal RNA and from ITS. Like most cases, this is not perfect, but it works out fairly well for a quick identification. For genera, remarkably, some genera can actually be resolved from a single gene, but that's not too often. And then families are still pretty perplexing for us from the data that we currently have, which is probably 5 to 6 genes at most, so we're waiting, obviously, for genomes. This is a tree that was put together from whole genomes. We have projects with JGI. A number of us in this room, as well as elsewhere, were involved in this project, and we sequenced the 16 genomes, which are highlighted in the dark font, and then combined with already published genomes. This tree by, let's see, I think Marcus generated, I believe, and Marcus Gerker, and it is tallies very nicely, or maybe I should say our other trees tally very nicely with it. You'll see no bootstrap values because they're 100%, which is often typical of these genome comparisons. But basically, whoopsie days. What I'm going to do, there we go. The reason for showing you this is that in my comments on the yeast, I'll be using another tree, one that we developed with 5 genes, obviously a lot less information, but we have a lot more species in it. So I just wanted to point out that basically our weaker trees do in fact track with this pretty well. And so our conclusions on those should be roughly valid. Probably the major points, we have some of the xylous fermenting species at the top, which come together very nicely. We believed all of these taxa were together, for example, from our earlier data sets, and again, the Saccharomycetaceae, which are represented by 3 species here, are nicely clustered. And then once again, Lipomyces, interestingly enough, seems to be the basal-most member of the Saccharomycotina, and interestingly, most of those species are from soil, as apparently their primary habitat. Now, one of the issues that face us all is the change in the code, and because of the prior code, we had lots and lots of yeasts that didn't form sexual states. And the simplest thing to do was just dump them into the genus Candida, which allowed us to develop a enormously large and polyphyletic group. Now, with the change in the code, we suddenly have nearly 500 species to reclassify, and the question is, what are we going to do? And I'm just going to touch on this briefly because Heidi Daniel will touch on it in much more depth on Friday, but the plan roughly is to combine the species in the Candida tropicalis, Lachancea and Longosporus clade as Candida. This will catch Candida albicans and some of the other medically important species. And so this will be considered Candida. Lachancea is named to be rejected and reclassified as Candida. And then all of the other Candidas will be placed elsewhere. Quite a number of them actually can be incorporated into existing genera, but on the other hand, we have quite a few that will end up in new genera. And so I'll just sort of touch on some of those. Now, this is the larger tree that I was telling you about that pretty much tracks with the genome tree, and I'll just try to point out where we seem to have problems. Once again, the limits are, here is the Saccharomycetaceae, then the Saccharomycotaceae right here, which kind of got fused into this clade 1. The major problem we saw as we deliberated on this group is that this genus Kazakhstania, which I put together and recognized as pretty large and pretty loosely supported, has now about doubled in size, and we all look at that and we see moderate basal support, but everybody is thinking surely this is going to be broken down into several genera in the future. The other point I'd like to make from this is that many of these genera are very difficult to separate phenotypically, which is, I think, what we're seeing is a pattern among many fungi. Syneclomyces, for example, at least is distinctive in it has unusual growth characteristics. It requires enriched CO2 atmosphere. Some differences, for example, Saccharomyces species tend to be high alcohol producers. Eremothecium, I'll mention a little later, produces very unusual ascospores in contrast to the rest of this group, but definitely trying to identify these just from phenotype can be just about impossible, and of course that's why we had all these reorganizations. Among this group, probably notable is Ascoidea, which indeed is distinct in terms of phenotype, similar to, well, the new genus Ascoidea looks much like it. Very distinct ascosporic state for Pachysolon with the little elongated ascophore. It looks like a little flask coming up, flaring, and the ascospores are in there. But beyond that, phenotype is not so distinctive among these. There are a few exceptions that I'll talk about in just a minute. Ding dong. Okay. I want to just briefly show you Kazakhstani. As you can see, the base of that's about 76% bootstrap values with data from most of 7 genes, some pretty small, but nonetheless, this was a fairly large data set, so there's a lot of resolution yet to be done here, especially if you can add in now, think about 30 species. Now that we think we have resolved the Candida problem, that it should even please the medical mycologists, we will present them with another problem, and that is Candida glabrata, probably the second most commonly isolated yeast in clinical labs, can no longer be Candida because of the change in rules once again. And there are a couple other species that are also medically important in this little clade of Nockaceomyces. So probably our strategy will simply be place them in Nockaceomyces, and if people want to call them Candida, so be it. Now, just touch on morphology briefly here. This is typical of the Saccharomycetaceae. This one lineage is really kind of interesting. Lachancea, Kluyveromyces, and Eremothecium. For most of the whole family, we have rather small spherical or sometimes reniform ascospores. For example, with Kluyveromyces, those particular spores are spherical. Lachancea, Sidri, for example, has similar spores, but now we get into Eremothecium, and this is really remarkable. Very large asci, multi-spored asci, depending on the species, either a very long thorn-like tail on the spore, or even in some cases, the old Nematospora, a long whip-like spore. So I'm kind of amazed by the fact that there's so much difference for a single genus here, but repeated data sets have shown us that this seems to be properly placed. Now, another one, a genus that has a rather unique phenotype is that Ambrosiozyma. The hyphae have large dolopore-like septa, similar to the dolopores in Basidiomycetes. This is unique for the yeast. And this has been recently expanded by adding these candidas. Unfortunately, they don't produce true hyphae, or true hyphae at all, but nonetheless, in terms of analysis, they seem to belong here, so that removes several candidas from the situation. And the same is true for Saturnispora. Genus is well supported. This is just a D1, D2 data set, but our more extensive data sets tell us the same thing, that good support, and so we've basically transferred as new combinations some of these candidas into Saturnispora. Then we had this persistent Candida abesophylla, which persistently ended up as initially a lone lineage, a single species on a long branch. Finally, Candida asiatica came along. And so I just recently proposed naming those as Martiniozyma, a separate genus, and I think we're going to see more of that. The other proposal that came out of our meetings the last couple of days was to reject Decra, an ascosporic name, in preference to Britannomyces, which is much more commonly used. So we'll see if that's approved. And then the genus Ogataea, which is of interest because the species will grow on methanol. And what we have found here is that they actually split into two clades, not much support to join them. So are we looking at sister genera? And so we had a bit of a discussion about that, and maybe a stronger data set will allow us to combine or definitely separate these two clades. Meanwhile, I think everybody would feel confident about transferring the few candidas in this lower clade to Ogataea because the type species is here. Okay, this is kind of the middle section of my Ascomycete tree, and in terms of the discussions we've had, we seem to have essentially no controversies. We believe most of these genera are fairly well circumscribed. There are a few large, especially one large genus that we'll hear about, Andre Lachance, on Friday, and I think the rest of them, for the most part, are fairly well circumscribed. Saccharomyces spartani, which we knew was probably misclassified, will ultimately be moved out of, because here's the type species. But this group is fairly stable at the moment, we believe. And then finally, the lower part of our Ascomycete tree, and some new genera to account for these long lineages of candidas. One here, Middlehovenomyces. Spencer martinsiella was recent. Denziella was fairly recent. Trichomon ascus, probably by our consensus in the meeting, will end up to be Blastobotris, the anamorph name. And I think, and then Lipomyces, I'll talk about in a minute, has some problems as well. One of the things that we were able to do is to For a long time, I think we all knew that Schizoblastosporion was really a member of the Nadsonia clade, but the old code prevented us from combining those. Nadsonia has priority, and so that was recently transferred. Lipomyces is probably our problem child, one of them. The Lipomycetaceae always end up in the analysis highly supported on at the basal node, but the interior branches show really pretty much weak support for a lot of these clades, subclades. There have been some proposals to name each of the subclades as a distinct genus, which may be correct. I'm not too excited about that without better data. This one was based on small subunit, the entire large subunit elongation factor and mitochondrial small. Clearly not enough to resolve this, so that's in the offing of something we need to deal with. Then I'll just kind of finish on the Saccharomycotina quickly with this illustration. First off, we have two distinct clades in this analysis, which we expect to probably hold. We have the issue that ecologically they seem similar. Do we combine them, or separate them because they clearly are separate clades, or accept the fact that we've got a deeply divided genus? That's under discussion. The other aspect in the little dark boxes are assay results on sophorolipids, and a number of the species in this upper clade, the Starmarella clade, or Starmarella because a few of the ascosporic states are here, are common. So one of our people was interested in sophorolipids, knowing that Bombicola and Apicola produced sophorolipids. We looked at some of these others and indeed found additional species. So that might be an example where we can actually help biotechnologists by using our trees. Now, the Taphrinomycotina is a very interesting group. As you know, it's probably the basalmost lineage in the Ascomycota, appears to be, and yet there are less than 200 described species. I think from the earlier slide we saw, as of 2008, there were 153. So I ask myself, why on earth can something be around this long and have so few species? Well, as I think you'll hear on Friday, and I've known from earlier papers, at least for one genus, the Archaeorhizomyces, that's about to change. But I've borrowed the tree that was published by Menkes et al. Just not so very long ago. It was a very nice tree, and it nicely illustrates the arrangement of genera within the Taphrinomycotina. And interestingly enough, we have fission yeasts, Pneumocystis being apparently dividing by fission along with Saccharomyces, a well-known fission yeast, sharing a common lineage. Then the plant pathogenic species, obviously grouped. Cytoella we don't know about at this point, but may well be a pathogen, a plant pathogen somewhere. And then this group that seems to represent perhaps mycorrhizal fungi or some association with living plants. Now, this is a lot of genera. The A's are anamorphs, if you will, and the T's are teleomorphs. And there is a bit of a problem with some of these because they have been reported only a few times. And let me just first, I'll kind of sum that up in the next slide, but I did want to point out on cell division, here's an example of protomyces, polar budding, for example, not so atypical of basidiomycetes, the same for Taphrina, an example of fission. And the interesting observation from Sugiyama's lab that enteroblastic budding was the norm for protomyces and Taphrina, but instead we have the typical ASCO2 layered cell wall, and it's also diazonium blue B negative. So is that enteroblastic budding a carryover from the common ancestor of Ascomycetes and Basidiomycetes? We don't know, but it's interesting. Now, other problems we have, What are the genera in red? Barenia, Protomycopsis, Taphridium, and Volcaria. We have herbarium specimens for those, but we have no living cultures, no sequences. Are they in fact misclassified protomyces? You know, we don't know. So this would be an interesting project for somebody if they could retrieve DNA from herbarium specimens. But in any event, with the exception of Pneumocystis, the rest of these are either plant pathogens or somehow associated with plants. So I think this would be kind of a super group to look at, but maybe very hard to deal with. With that, thank you very much.
Thank you, Clete, for your nice overview of Ascomycete yeasts, and I think there is time for one short question.
Uh-oh.
You were talking about Basidiomycetes similarities with was it Protomyces and Taphrinomycotina?
Yes.
But I just wanted to point out Saccharomyceta is, I think, positive, Diazonium B positive.
Which one?
Saccharomyceta.
Oh, no, it is not.
It's not?
It is
You're
not.
sure? Because that was
I am
in the
sure.
original, it's in their book from way back.
Yeah.
But that's wrong.
It should be, it should be negative, I think.
And you've tried it again on the,
I think
it's in the literature, though,
As
that
a
it's
positive?
positive. Yeah,
Yeah.
so I'm just curious about that.
Well, I may be mistaken, but I'm almost positive.
But
it's
your
negative.
new species was,
I
you
did
would,
not run it.
Okay.
We only just identified that from gene sequences.
okay.
Well, Clete, maybe I have a very brief question. What will be the fate of Candida?
Beg your pardon?
What will be the fate of Candida?
The fate of Candida?
The name Candida, yeah.
That was on that early slide in which, pretty much as we discussed in our workshop, it'll be circumscribed around Tropicalis albicans group. And you will get a much more complete story that Heide will give you on Friday. So I'm trying to drum up business here for her talk because she's got a lot of nice slides and a lot of nice analyses. So be sure you go to it. And the same, of course, for the talk on Archaeorhizomyces, which should be really a fun talk.
Okay, mind steps.